Once we have checked the quality of our reads and trimmed them if necessary, we are ready to proceed to aligning the reads to the reference genome. Here the goal is to find out from which location the reads originated from. But there are a couple of challenges. So our reads obviously contain genomic variants, and they can also contain sequencing errors. On the other hand, genomes have non-unique sequence and introns, while our reads come from mature RNA where the introns have been spliced away. So the RNA-seq aligner needs to be able to map those splice junction spanning reads to genome non-contiguously. And this is difficult because, well, while they are splice signals, they are relatively weak. And on the other hand, introns can be thousands of bases long. So in this image here, we have a piece of genome, a piece of gene with 3 exons, exon 1, 2, and 3. And then we have different kinds of RNA-seq reads. So the red read here maps within an exon, and that's easy for the aligner. Then we have other reads which actually go across exon-exon junctions, so they need to be mapped non-contiguously. And the ones that are particularly hard are these ones which have a very small overhang in the other exon. So there are lots and lots of alignment programs developed. Typically, they take the genome FASTA file, and before doing any alignment, this FASTA file actually needs to be converted to a data structure which is more efficient for searching similarities. So some of these aligners use so-called Burrows-Wheeler transform index, and some are based on suffix array approach. The aligners differ in their speed, also in their memory requirements, accuracy, and the ability to deal with spliced alignments. So when you are mapping RNA-seq reads, make sure that you use a splice-aware aligner. There are several around, but probably the most known ones are these. So there is a STAR, which is very fast and accurate, but it needs a lot of memory when doing the alignment. There is HiSet2, which is also fast and accurate, which doesn't need as much memory as STAR, but when it creates the indexes, it actually needs a lot of memory. And then there is TopHat, which is kind of the previous version of HiSet, which is a bit slower and it needs less memory. So in Chipster, we have these 3, but there are differences in what genomes are available on the Chipster server indexed for these particular aligners. So for STAR, we have the human genome index available. For HiSet, there is human and mouse. And you should let us know, of course, if you want to use others, so we can make those indexes. HiSet tool in Chipster also offers the possibility that you can supply your own genome. But here, if you do so, the genome should be relatively small because, like I said earlier, making the genome index consumes a lot of memory. And for the TopHat, for historical reasons, we have many genomes available. And again, you can use your own genome there too. All of these guys produce similar output files. So they all produce a BAM file. In a moment, we will look in more detail what BAM file structure looks like. But basically, BAM is the file that contains the alignments. Then there is an index file for BAM called BAI, which is required for genome browsers and also some analysis tools. The log files produced by aligners can be very useful in order to check how the alignment run went. So let's start with HiSet2. So the name HiSet comes from these words. And like I said earlier, it's a very fast aligner. And its memory consumption is relatively low. So the reference genome is indexed for fast searching. Currently we have these two indexes, but you should let us know if you need others. And you can also, of course, use your own reference genome if you want it to, as long as it's relatively small. Now, the way how HiSET2 works is that, importantly, it actually has two types of indexes. So it has a global index, which is used to place the read in a genome. So as long as there is a 28 base pair stretch which matches and matches uniquely in the genome, we place the read in that region. And then HiSET uses another smaller index for that particular area to do a rapid extension of this alignment. Now, this approach, this two-level approach, is particularly good for those reads which have very short overhangs over the splice sites. The other smart thing that HiSET is able to do is that as it's running, it encounters splice sites, it keeps those in mind. So when it takes the next read to align, it can use the information it collected from the previous reads. Again, this is good for those splice reads with very short overhangs. If we look at the parameters for HiSET2 in Chipster, so you obviously have to select the genome. It's also important to indicate the library type, which refers to the strandedness of the data. As we discussed before, RNA-seq can be made with a stranded protocol, and there are different ones for those. Then by default, HiSET reports maximum 5 alignments, but for a read. But it's good to remember that that they are not necessarily the absolutely the best ones. Then it's also important to indicate what base quality encoding was used. So if your data is relatively recent, then it's probably the CIGAR encoding or PRED plus 33. The other things you can set is you can require long anchor lengths if you are planning to do assembly later on. So then you only allow alignments where the splice site overhangs are at least 16 bases or longer. Then let's look at the star aligner. So the name star comes from these words. Star converts the reference genome to a suffix array for fast searching. And the star in Chipster uses a 2-pass protocol, which is useful in that in the first pass, so it collects the splice junctions that it discovers, and then it actually adds those splice junctions to the genome index and remaps all the reads again, and this is the second pass. So this approach, while it doesn't actually increase the number of new splice junctions, it allows more spliced reads to map to these novel junctions. Then differently from HiSET, star has a parameter which says maximum alignments per read. And here the meaning is that if a read has more alignments, more equally good alignments than this number, then none of them will be reported. If it has less alignments than this number, then all of those alignments, if they are equally good, will be reported. Chipster has some ensemble GTF files on the server, so you can give this information, which contains annotated splice sites to star, but you can also give your own. So, for example, GenCode GTFs are recommended. Now, you might end up in a situation where your sample has actually several FASTQ files. So, depending on the sequencing technology that was used, it could be that you have, for example, 8 FASTQ files per one sample. In this case, it's important that you align all of them together in one alignment run. If your data is single-end, you can just simply select all those FASTQ files and run the aligner. If your data is paired-end, then you need to make an additional step first. So you need to make file name list files. And the way how you do it is that you start, so in my example here, for example, we have 8 files. So these are the sort of forward reads, which have we call them read ones, and these are the reverse ones or read two. So what I would do is I would select first all the read one files and run a tool in the utilities category called make a list of file names. So this will produce a list which just lists the file names of these. Then I would repeat the same for the read2 files, so I end up with 2 file name list files. And then I go to HiSet2 or star, so that I select all the FASTQ files and both these file name list files. And when I go to the parameter settings of the aligner, in the end of parameter set, there is this section called input datasets. And here I want to make sure that I indicate the FASTQ files that they contain the reads, and then I assign the file name list files correctly so that the read one files are listed in this one and the read two files in the other one. And once I'm ready with this, then I can run the aligner and all the FASTQ files are aligned correctly in one go. So the reason for having to make the list files is to make sure that these read1 and read2 files are paired correctly. Now let's have a look at the result files then. So the alignments are in the BAM file. So BAM is a compact binary file containing the aligned reads. As it's a binary file, you normally cannot actually look at it, but Chipster has a BAM viewer which allows you to view it as text. As it turns out, BAM is actually a binary format of a SAM file, so that's where the name comes from, sequence alignment map, and BAM then is the binary of this. And the SAM file contains exactly the same information as BAM, but it's in a tab delimited text format. So this file would actually be human readable. It's just that they tend to be very, very big. So it's preferred to store the alignment information in the BAM format. Now, if we have a look at the BAM structure, so the BAM files start with the header, which goes up to here. And the header contains various things. So there is, for example, information of whether this BAM file has been sorted and how. So you can sort this based on read names or genomic coordinates. Then we have a lot of rows here. These are actually the chromosomes. So reference name, sequence name, and length. Then there is a program line which tells which program produced this BAM file and what was the actual command that was run. So this section is very useful because if somebody gives you a BAM file and forgets to tell how it was produced, you can always check it here. Now, after this header section then comes of tens of millions of rows, and each row refers to one alignment. So like here now, I have 4 rows, but this is actually just because the text has been wrapped. So all this information now would normally be in one line. So here we have the read name, then various pieces of information like to which chromosome this read aligns and which position and so forth. Then we actually can see the read sequence and the quality string that we discussed earlier. And then we have a whole heap of tags afterwards. Now these fields always come in the same order, which is listed here. So read name, a thing called flag, which we will talk about soon, name of the chromosome and the position, mapping quality, and CIGAR, again things that we will soon discuss. And in the end, you have tags. The number of tags can vary. Some are general to all aligners and some are specific to a particular aligner. The tags contain various information, but an important one is an interesting one is this one. So NH tag, for example, tells you to how many positions in the genome this particular read aligns. So here it says that this read maps to 7 positions in the genome. There's actually a very nice website if you want to read more about this BAM structure. But let's look at some of these things. So we have mapping quality and CIGAR and the flag. So mapping quality tells us how likely it is that the read really comes from that position where the alignment has placed it. So before we discussed about base quality, which was the confidence that the base call is correct. Here we have mapping quality, which is the confidence that the alignment position is correct. This depends on many things, but the most obvious one is how unique that particular place is in the genome. So, in other words, if a read maps equally well to several places in the genome, we cannot have any confidence that it comes from one particular one of them. Mapping quality is expressed in PRED scores, just like the base quality, so this formula is the same. But here the mapping quality is shown in numbers, so it's not converted to those ASCII characters that we discussed in the context of base quality. However, things are not quite as clear-cut, so different aligners actually have slightly different ways of indicating, for example, a unique mapping position. So for hazard2, that value is 60. For star, it's 255. And in top hat, it's 50. And there's actually a very nice blog post about this issue. Then one thing that you have in the BAM file is the CIGAR string. Now, what does that mean? Well, essentially, it tells you how the read maps in that position. So CIGAR consists of numbers and letters, and the letters have different meanings, like indicated here. In our example here, we have a very small reference sequence, and then we have our read. So let's look at the CIGAR. So it says 8M, so you can see that there are actually 8 matches. Then 2I, so 2 insertions. The read has 2 bases which are not present in the reference. Then we have 4 matches, And then we have one deletion. So the G is missing from the read. So 1D in the CIGAR. The flag field in the BAM file is a number, and it's actually a sum of different values. So it's a very compact way of storing information. There are websites which can interpret these numbers. Here we have one screenshot. So, for example, flag 403 would tell you that the read is paired, it's mapped in proper pair, is from reverse strand, it's second in the pair, and it's not primary alignment. In addition to the BAM file and the BAM index file, you also get log files, and this can be very useful. So here we have an example from the high-set aligner. So the information you want to check here is that, well, how many reads actually mapped to the reference and how many of them mapped uniquely. If it was paired data, you can check how many pairs mapped and how many of those pairs mapped concordantly, meaning in the right orientation. And importantly, what was the overall alignment rate? So like in this example here, the overall rate was 82.6%. Here is another example. So this one is from Star Aligner. Here you can get a bit extra information also regarding the splice sites, for example. Then there are also other tools. So the ones that we looked now were just the log files produced by the aligners, but there are also other tools for checking the BAM files. These live in the utilities category in Chipster, and they are typically based on the SAMTools package and the Picard package. So you can count how many alignments there are in the BAM. Here I have underlined the word alignments to remind you that there are always going to be more alignments than you had reads to start with, because remember, one read can have several alignments. There is also a tool checking how many alignments in the different chromosomes there are, and so forth. There are also tools for manipulating BAM files. So you can make a subset of BAM for your favorite chromosome or region. And this subsetting can also use mapping quality. So you can take only the good quality mappings to that region. When you run an aligner in Chipster, you will always get the index file. But if you get a BAM file from elsewhere, you might not have the index. So there is a tool in the utilities category which can produce the index for you. And actually, if you are importing a BAM file to Chipster, it will ask you, do you want to pre-process it? If you say yes, it will sort the BAM file and index it. So sorting here means sorting based on genomic locations. If you are importing a SAM file, it will also convert it to BAM. Now, this pre-processing question, when you ask yes, this pre-processing will happen on your computer, but it's perhaps better to say no at that point and then run a tool in the utilities category, which will do this, because this tool will run on the server side and not slow down your computer.